Namaste friends, welcome back to Biology Center for True Learning. In this video, we discuss about one prominent person, the important geneticist Th Morgan. Th Morgan, Thomas Hunt Morgan. He is a father of experimental genetics. He is also called flyman of genetics. Flyman of genetics because he selected Drosophila Melanogaster, which is also called fruit fly, for his experiments. Because of selection of fruit fly for his experiment, Morgan also regarded as flyman of genetics. The Drosophila Melanogaster, so also regarded as the jackpot of genetics. In the year 1910th year, the Th Morgan suggested that both the aspects, copulation and repulsion, are the same aspects of the concept linkage. Firstly, the linkage phenomenon was seen by W. Bateson and Pinnett. But originally, the linkage concept was verified and proved by the Th Morgan and his colleagues only. Right. The Th Morgan proposed the theory of linkage. The theory of linkage was given by Th Morgan. So let us see. So what are the principles said by the theory of linkage one by one. The first one, many genes are present on chromosomes. Each chromosome possesses so many genes. Second one, genes located in a linear fashion. Genes are present or located in a linear fashion. Next, third one, genes present are linked each other. Genes present on the chromosomes are linked each other. Linked genes inherit together at the time of meiosis. Linked genes inherit together or transmitted together from generation to generation. Next, so another point, the fifth point, strength of linkage. The strength of linkage depends upon the distance between two genes. Right? When distance is wider, I mean there is a long distance between two genes, linkage is weak. For example, this is the chromosome. So here is one gene is present, here is another gene is present. Here the gap is more, the gap is wider. So the linkage is weak in this case. Then when distance is closer or shorter, then the linkage become strong. So again we take another example. This is the chromosome. Here two chromosomes are very closely located. These are strongly so attached. Here the linkage is strong. Next, law of independent assortment is not universal. It is not universal. So another one, linked genes do not show law of independent assortment. The linked genes do not show the law of independent assortment. Two dominant genes or two recessive genes present on the same homologous chromosome. That arrangement is called a cis arrangement. So this is like that. So this is the chromosome. So whether both the dominant, both the A and B, both the dominant genes present on the same chromosome or both recessive genes present on the same chromosome. This type of arrangement is called cis arrangement. Then one dominant, one recessive gene present on the same homologous chromosome. That arrangement is called a trans arrangement. So for example, this is a member of homologous pair of chromosome has one dominant gene, so another one is a recessive gene. So it is another chromosome has one recessive gene, one dominant gene. This type of arrangement is called a trans arrangement. Next, so this is about the theory of linkage. Next, so we are going to talk of the reasons behind selection of the Drosophila melanogaster as experimental animal. So there are many animals. Morgan only selected fruit fly for his experiments. So are there any advantages behind selection of this animal? Yes, there are some reasons, some benefits behind selection of the Melanogaster. Reasons for selecting fruit fly. The first reason, they can reared easily under laboratory conditions. So make them grow in the laboratory condition is easy. That is not so much of risk. One reason. The second reason, Drosophila has a short lifespan. It is about 2 weeks. The lifespan of the fruit fly is 2 weeks. The another reason, single mating gives rise to production of a large number of fruit flies. Then here the breeding is not seasonal. Throughout the year the breeding can be performed. Then another reason, they can grow on a synthetic medium. medium, so which is made available in the laboratory. This synthetic medium contains the yeast, then cream of wheat, then agar, then molasses. These are the nutritive materials available in the synthetic medium. So on that synthetic medium, the fruit flies can easily grow. The another reason, they have four pairs of distinct chromosomes. Morphologically, these chromosomes are different. Next, seventh one, male fruit fly can easily distinguishable from female. Sexes are separate. Male fruit fly, female fruit fly. The male can easily differentiate from the female. Here the male is in small size, but female is in large size. Then we can easily identify by identifying the ovipositor. Ovipositor is the egg-laying structure present in the female fruit fly. Then another reason is the large chromosome polytene chromosome is present in the salivary gland of the larva. So that can the study of that chromosome helps in the identification of the chromosomal aberrations. Then the another reason for selection of so this one is okay, these are enough. So these are the main reasons. For selecting the fruit fly as experimental material for Morgan's experiment. Here, one more thing I would like to tell you. Morgan, Th Morgan worked on some important aspects, some important phenomena. The first phenomenon is linkage. He studied the linkage, then crossing over. Third one is sex linked inheritance. Another one is crisscross inheritance. The fifth one is linkage maps. Morgan studied these 5 phenomena separately. So here I briefly tell about each one. In further videos we will have a detailed discussion about each one. So what is linkage? The tendency of 2 or more genes to retain together during the inheritance process from generation to generation, that is called a linkage. The genes are linked on the same chromosomes remain together at the time of transmission from parents to offsprings, that is linkage. Then crossing over. What is crossing over? The interchange of chromosomal parts between non-sister chromatids of homologous pair of chromosomes is called crossing over. Then sex-linked inheritance means the transmission of body characters from parents to offsprings along with a sex is called sex-linked inheritance. Then what is crisscross inheritance? Here the inheritance of gene from mother to son or from father to daughter is called crisscross inheritance. Then linkage maps. So a map of genes prepared based on the linkage analysis. So after finishing the linkage analysis, it is possible to make a maps of genes. That kind of maps are called linkage maps. Thank you for watching this video.